Some animals alive today were already ancient when human civilizations were in their infancy. In each case, the biology that enables extreme longevity violates assumptions about how aging works, and researchers studying these species are actively extracting principles for human medicine. Greenland sharks, Somniosus microcephalus, reach sexual maturity at approximately 150 years of age and have a maximum confirmed lifespan exceeding 500 years. Radiocarbon dating of the eye lenses, which form from proteins produced before birth and never replaced, allows accurate age determination. The oldest specimen confirmed was 392 years old at minimum, with a central estimate of 512 years, meaning it was born no later than 1627. At that point, the telescope had been invented 4 years earlier. Greenland sharks grow approximately 1 centimeter per year. They are slow, live in near-freezing deep water, and have extremely low metabolic rates. Unusually, they reach sexual maturity long after most other vertebrates have died, suggesting reproduction does not accelerate aging as it does in shorter-lived species. Ocean quahog clams, Arctica icelandica, are the longest-lived non-colonial animals confirmed by direct evidence. A specimen collected from the coast of Iceland in 2006 and named Ming, because it hatched during the Ming dynasty, was determined to be 507 years old. Researchers confirmed its age by counting annual growth rings in its shell, the same method used to age trees. The act of opening the shell to count the rings killed the animal. Ming had been alive when Columbus reached the Americas and when Michelangelo was painting the Sistine Chapel. Ocean quahogs live in cold, deep water with very low metabolic rates. Their cells show exceptional resistance to oxidative stress. Bowhead whales, Balena mysticetus, live over 200 years. The evidence is not indirect. Traditional Inuit hunters have recovered 19th-century stone harpoon points still embedded in the blubber of whales killed in recent decades, meaning those animals survived the initial strike and lived for over a century with the point lodged in them. DNA-based age estimation places the oldest confirmed bowhead at 211 years, suggesting some individuals may live longer. Unlike most large mammals, bowhead whales show no age-related increase in cancer rates. Researchers studying their genome have identified multiple genes involved in DNA repair and tumor suppression that differ significantly from human equivalents. Antarctic glass sponges, Rossella antarctica and related species, grow in some of the most stable environments on Earth, deep Antarctic water at near constant temperature. Individual specimens have been estimated at between 10,000 and 15,000 years old using isotope dating methods. At typical growth rates of less than a centimeter per century, a sponge 2 meters tall represents tens of thousands of years of continuous biological activity. These sponges were alive and growing when humans first entered the Americas and before the last ice age fully retreated. Because sponges are among the simplest animals, they lack a nervous system, muscles, or organs. Their longevity appears to involve extremely efficient cellular repair rather than any complex biological anti-aging mechanism. The naked mole rat, Heterocephalus glaber, is the longest-lived rodent documented, with individuals confirmed at over 37 years, roughly 9 times longer than other rodents of the same body size. Standard mammalian lifespan scales predictably with body mass. Naked mole rats weigh 35 grams and should live 3 to 4 years. They do not follow the expected increase in cancer risk with age. Their cells tolerate extraordinarily high levels of reactive oxygen species without dying. They maintain cardiac function and reproductive ability into old age. A queen that has reached 30 years can still breed successfully. Researchers have not fully identified the mechanism but have noted unusually stable ribosomal function and high levels of proteasome activity, better than average cellular garbage disposal. Hydra, Hydra vulgaris and relatives, are freshwater polyps a few millimeters long that show no measurable sign of biological aging in laboratory conditions. Their bodies are composed almost entirely of stem cells, approximately 3,000 in a typical individual, which divide constantly and replace the entire organism's cell population every 20 days. A 1998 study tracking 145 Hydra for 4 years found no increase in mortality rate and no decrease in reproductive rate with age, meeting the formal biological definition of non-senescence. The protein responsible, FOXO, a transcription factor, is also present in humans and has been associated with exceptional longevity in centenarian populations. Hydra can be killed by physical damage, infection, or starvation, but not by age-related deterioration. Tortoises hold multiple longevity records among land vertebrates. Seychelles' giant tortoises, Aldebrachles gigantea, have documented individual ages exceeding 190 years. Jonathan, a Seychelles tortoise on the island of St. Helena, is estimated to be at least 192 years old as of 2026, making him the oldest confirmed living land animal. He was present when the photograph of him at an estimated age of 50 was taken, in 1886. His diet has been hand-supplemented by the island's governor. He is partly blind and has lost his sense of smell, but remains otherwise functional. Tortoise longevity is associated with slow metabolism, low body temperature, and exceptional DNA repair capacity. Their telomeres, the protective caps on chromosomes, do not shorten at rates comparable to shorter-lived species. Flat oysters, Ostrea adulis, and other bivalves demonstrate that extreme longevity is not confined to exotic species. Age determination via shell ring counting has confirmed multiple flat oysters at over 40 years and ocean quahogs routinely at 200 to 400 years in undisturbed populations. The pattern across long-lived animal species is consistent: slow metabolism, cold environment, exceptional DNA repair, and resistance to oxidative damage. Researchers studying these species have identified multiple molecular pathways: mTOR inhibition, sirtuins, proteasome efficiency, and FOXO transcription factors, all of which are present in humans and all of which have been associated with extended lifespan in experimental conditions. No single mechanism accounts for extreme longevity. It appears to require all of them simultaneously. These animals are not curiosities. They are working biological systems running successful longevity programs that researchers are now attempting to replicate. Tomorrow's fact is the one most people can't believe is real. This channel posts a new animal fact video every day. Subscribe so you don't miss the next one.